Auzubillahi minash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. Rabbi zidni ilma. Okay, the today's lesson that we are going to talk about is the cell cycle and the cell division. And before going into the details of cell cycles and its different type of cell divisions, first of all, we need to identify, or we need to talk about certain terms that we are going to use here, as number one, daughter cells, number two, parent cells. There will be chromosomes, chromatin network, there will be haploid cells, diploid cells. So multiple things are going to be used as a terminology while studying this type of cell division in detail. First of all, it says that when a single cell divides to form two identical daughter cells, the condition or the phenomenon is called as cell division. The new cells which are formed after the process of cell division is completed are called as daughter cells, and the original cell from which the daughter cells have arisen, or the origin from which the daughter cells have been produced, is called as parent cell. Hence, one parent cell undergoes cell division to form two daughter cells, and this is the normal process of what we call as mitosis in our daily life. Then the next thing you can see here is one single parental cell which is actually going through the division process, and the multiple steps are there. When the process begins, it continues, and when it's finished, when it's done, finally two daughter cells are produced. And here you can see the two daughter cells which are produced are identical as the parent cell. Just one difference you can see here is the daughter cells which have just been formed are slightly smaller in size as compared to parental cell, but after some time these daughter cells will get mature into a full-fledged complete cell which will be exactly same in structure and shape as the parental cell. Then the next process of cell division will begin. Talking about cell reproduction, we mainly have two major things behind that. The first one is the type of asexual reproduction, in which the gametes or sex cells are not involved, while the second main thing which requires cell reproduction is the sexual reproduction, in which the gametes or germline cells are actually involved. When I'm talking about the asexual reproduction, that means which is not actually done with the formation of gametes, or it has nothing to do with some sort of fertilization and all that stuff. It can be binary fission and it can be mitosis. Binary fission is a simplified form of mitosis which actually occurs in case of small unicellular organisms like bacteria, etc. While in case of eukaryotic cells, a proper formation of new cell occurs through passing through multiple stages or multiple steps and phases, and this complicated process is called as mitosis. As a result of binary fission and mitosis, two cells of exactly same parental type are produced, but the difference between binary fission and mitosis is actually the phases difference. So binary fission is just a simple step in which single cell simply divides into two by just getting converted into physical states, while in case of mitosis, multiple steps are involved. While in case of sexual reproduction, when cell reproduction is considered, I'm sorry, that is a specific type of cell reproduction called as meiosis, in which the cells which are formed, the daughter cells which are formed from parent cell, are actually having half the number of chromosomes and are 4 in number, while in case of mitosis it was 2 cells produced, but the chromosomal number was same as the parental cell. So that will be the difference, inshallah, that we'll be studying in the next classes. So here you can see there's binary fission, and you can see the example of paramecium, and this is a unicellular organism which lives alone, which lives a solitary life. In this case, you can see the organism's nucleus is first going to be divided into two, and finally the cytoplasmic content is being divided. And finally, after completion of the nuclear division and cytoplasmic division, two individual cells are made just from one single cell. So this process does not require any specialized steps or phases, but in case of mitosis, we have to pass through different phases, which will be, inshallah, studied in our later part of lecture. So in mitosis, generally we have first of all DNA replication, and after replication the mitosis process begins, and finally two diploid cells are actually produced, which are having exactly the same number of chromosomes as the original parental cell. While in case of meiosis, you can see we started with position A, and after going through the first meiotic division and the second meiotic division, finally we have got 4 daughter cells which are actually having half the number of chromosomes as compared to the original parental cell. So one piece from each chromatid was chromosome was actually got into the one single cell. So these are called as haploid cells because they have half number of chromosomes as compared to the original diploid or parental cell. Now starting the going towards the packaging of DNA. DNA is present in the nucleus of the cell, as we all know that, and it is something which is condensed, which is having actually occupying the smallest portion of your cell, that is the nucleus, and it is highly convoluted and condensed level. So it fits inside the nucleus, and the DNA is normally present in the form of chromatin, but when the cell is going to be divided, it gets more condensed, it becomes more visible, and then we call it as chromosome. So chromatin is actually the type of DNA which is not in the active state of division and is invisible and is thin, while in case of chromosome, this is the form of DNA which is visible, which is thicker than chromatin and is visible during cell division only. Thus, DNA is tightly packed into the chromatin, and chromatin is tightly packed into the chromosomes. Here you can see we have this is the DNA. DNA has different type of proteins and called as histone proteins. Then the chromatin fibers are there, and finally, if you are going to make it condensed and in a full-fledged shape, we can have this X-shaped chromosome, which is actually 2 arms attached at the central point called as centromere. Here we have a detailed picture of structure of chromosome. You can see we have 2 arms, and these arms are called as chromosome arms, or simply called as chromatids. So one-sided chromatids are called as sister chromatids, and the opposite side chromatids are called as non-sister chromatids. So similarly, for example, we have named as P and P, Q and Q. So P to P are sister chromatids, Q to Q are sister chromatids. But if I'm talking about Q and P, they are called as non-sister chromatids, and they are actually attached to the central point called as centromere. And on the sides of centromere, there are 2 protein points called as kinetochore protein points, which are actually the attachment sites for the purpose of detachment of these chromatids during cell division, when the chromatids get apart and move towards opposite poles. So this is the detailed structure of the chromosome, and the last or the marginal ends of the chromatids are called as telomeres. Cell cycle involves 4 phases: the growth phase, in which the cell grows in size, that is G1 and G2 phase; the synthesis phase, in which the DNA multiplies or replicates, called as S phase; and finally the mitosis stage, or the cell division stage, in which the parent cell divides into 2 daughter cells, called as M phase. So M stands for mitosis, S stands for synthesis, G stands for growth phase. So order of phases are G1, S, G2 and M. Going into the further details, we have a sketch of complete mitosis. Here we have, first of all, first growth phase, growth in normal metabolic roles. So in G1, simply metabolism occurs. The cells are just going to produce lots of energy for itself, because this cell is going to be divided and it needs to have lots of energy for production of lots of energy. That is going to have more and more mitochondria, and more enzymes are actually going to be required. So it is actually growing its ribosomal numbers as well. So number of ribosomes, number of mitochondria are going to be increased. Metabolism is going to be increased in case of G1 phase. And after G1 phase, there is S phase. In S phase, the original DNA replicates. So actually the number of DNA which was originally present is going to be doubled in S phase. After the S phase is completed, the third phase is actually the second growth phase or second gap phase, and that is the phase in which the cell is actually preparing itself for mitosis. Spindle fibers, etc., proteins are going to be produced in here, and these are special proteins which are going to be produced, or which are going to be used for the formation of spindle fibers, for the separation of chromatids during cell division. And finally, the M phase begins, which is mitotic phase, which is further divided into 4 phases, which is prophase, metaphase, anaphase, and telophase. First of all, G1 phase, as we have just stated, it's a type of interphase. Interphase is actually the stage between two cell divisions, or you can say the stage which is before real cell division occurs. So this is called as the preparatory phase, in which the cell is going to be prepared for the cell division. So G1 phase is the first growth phase of the cell. The cytoplasm and organelles grow in the cell, and it prepares the DNA for the next phase, that is S phase. The cell performs normal cellular activities during this phase. And S phase, which is also called as synthesis phase, a new DNA is synthesized from the original DNA, hence the DNA number doubles up in the cell. If we have a chromosomal number 46, that is going to be 92 in case of S phase. In G2, it is the second growth phase of the cell. The cell prepares itself and grows the required organelles for cell division. As we have just discussed, the cell is actually going to increase its proteins, its mitochondria, and its ribosomes, which are actually going to be required for the purpose of cell division. And finally, we have got the M phase, which is mitosis or mitotic phase, which is the real cell division, and the parent cell is now going to be divided into 2 daughter cells. Each daughter cells will have the same number of chromosomes as present in the parental cell. How? That is what we will be inshallah discussing in details of the mitosis. For example, if parental cell has 8 chromosomes, then each daughter cells will have 8 chromosomes. The G1S and G2 phases are together grouped under interphase, and interphase is the duration between every successive mitotic division. So when the cell is successfully done with the first mitotic division, it now goes into the next interphase, and it prepares the new daughter cells which have just been formed to start another cycle of the growth, or the other cycle of the reproduction. So here we are done with the interphase. Now inshallah, in the next lesson we are going to discuss about the mitosis in detail.